Welcome to our final lecture of plant biology. Today we are going to be talking about plant reproduction and all of the parts that plants need to reproduce. So first we're just going to have this here, label it, tell you what each of these parts actually do, and then we'll get into the actual types of reproduction. So we're going to focus on angiosperms, which are flowering plants. In this specific example, we're talking about literal flowers and how they reproduce. So I'm going to throw up the labels and then we'll go through them. So this would be a really great picture to copy down into your notes. You should label them. And you should be comfortable being able to label an image if this were blank. So let's get into what each of these do. So you've got your functions on the right. We're going to match them up now. So you have your petal, which attracts pollinators. Petals are typically brightly colored. They smell good. The whole point of a flower's petal is to attract someone to, whether it's a bee or other animals. They want them to get the pollen and bring it somewhere else to pollinate, right? Flowers cannot move. Petals are neither male nor female. The anther, that is that very top portion right here, that is male and it contains pollen. The filament is kind of a long tube that connects the anther to the rest of the flower, also male. The sepal is a cover or protection that exists when the flower is developing and then turns into little green leaves. Neither male nor female. The ovary contains ovules, which are eggs, female. The stigma is this very top part right here. It is the pollen landing site. It is female. The style is the tube that grows from the stigma to the ovary and particularly the ovule. Female. And then the ovule contains the egg nuclei. Female. Now, process of reproduction has a few steps to it. So you have pollination, fertilization, and then seed dispersal. Pollination is the act of pollen being carried from one anther of one flower to the stigma of another flower. So from male to female. Male of one flower, stigma of another. Or male of one flower, female of another. Fertilization is different than pollination. Fertilization is the pollen tube grows down from the stigma to the ovary and the pollen is actually delivered and fertilization occurs. So fertilization is pollen hitting ovule. Pollination is pollen moving from flower to flower. And then seed dispersal is the idea of seeds being dispersed. So once the seed is developed in the ovule, it's ready to move to a new area, be pollinated again, planted so that it can grow, whatever it is that the plant we're looking at does. Now, again, as I mentioned previously, there are dicots and monocots, but we pretty much exclusively talk about dicots because that's just where most of the plants that you know and can associate with. Most of them are dicots. So for a dicot seed, there are a few major pieces that you need to know. On the outside, you should know the testa, the micropyle, and the scar. And then on the inside, you should know the plumule and the radicle. So on the outside, the testa is just literally the outside, the seed coat. Micropyle is a little tiny hole in the seed where water enters, and then the scar is where this seed was attached to the ovary. And then on the inside, you have the plumule and the radicle. This is the embryonic stem and root. So it's the very first stem and root that grows from the seed. Remember that I said dicots have 2 cotyledons, which are leaves. So from the plumule, 2 leaves will grow. Germination. I know you've done at least one germination experiment, but probably many germination experiments in your high school career. And you know that germinating seeds really comes down to having a few good conditions. Water, oxygen and CO2, ideal temperatures and pH, and light. The basics, all kind of living things need to survive. Same with seeds. But some seeds have really specialized conditions. So Klein grass, for example, needs to be digested and passed by cattle. Could be by anything, but cattle specifically do this before they're able to grow. Some need to be have their inhibitors removed by maybe rain, or in the water. It gets brought from one side of a river to another. And some of them, like the Cistus and Canis, which is what that picture is right there, their seeds need to be induced by smoke. So after a fire, when smoke is remaining, the smoke will allow the next step in that plant's hormones to kind of awaken, and then they'll be able to grow. So along with the basics that plants need to germinate, some have additional conditions that need to be met before they can germinate. Beyond the basics, you will need to know very, very specifically how starchy seeds germinate. I'm going to walk you through this, but I would suggest either drawing this or taking a picture from the internet and printing it out and putting it in your notebook, but you should definitely be able to tell me exactly how starchy seeds germinate. So, step 1 is water comes through the micropyle, right? That little hole in the seed, and activates the cell. A seed can stay in a dormant state for a very, very long time, and when water comes in, that's when all of the other hormones and enzymes and bits of a seed are going to kind of come to life. So once water is absorbed, gibberellins has become synthesized, right? The synthesis of gibberellins happens. We kind of talked about this when we talked about auxin in the last video, but gibberellins is just a plant growth hormone. So water comes in, gibberellins is synthesized. Once that gets synthesized, it causes the synthesis of amylase. Amylase is an enzyme, and once amylase is synthesized, then it breaks down the stored starch. Remember, we're talking about starchy seeds. So it breaks down the stored starch from starch to maltose. Once the starch is broken down into maltose, it's absorbed, the maltose is absorbed by the plumule and radicle. Once that happens, maltose gets broken down even further into glucose. And once you get to glucose, then you can actually use it for the respiration in the tissues that are growing in the plumule and radicle, which allow the plant to actually begin growing. So again, water's absorbed, it causes the synthesis of gibberellins. Once gibberellins is activated, it causes the synthesis of amylase, the enzyme. Amylase breaks starch down into maltose. Maltose is used by the plumule and radicle. And then once the plumule and radicle absorb the maltose, maltose breaks down again into glucose, and glucose is actually used to continue the growth of these starchy seeds. Okay, now, I, of course, saved the best for last for you guys. This topic, this idea of flowering in plants, is complicated. I have quite a few slides on it, and a lot of it is repetitive, but you will find yourself hopefully not totally lost after the first slide. But I want to warn you that this is a complicated topic, probably the most complicated topic in Unit 9. So please, please, please pay close attention. So, when we're talking about flowering plants, we know that the purpose is to allow for pollination. All living organisms want to continue on, right? They want to reproduce. So flowering plants flower, right? They bloom so that pollinators can come and help spread the pollen. Seed dispersal happens, and we know that flowers bloom when suitable pollinators are available. A flower is not going to bloom in the dead of winter when they know that there's no bees around to help spread the pollen. That is a waste of energy for the flower. So it's not going to do that. With that being said, there are two types of plants. There are plants that bloom in long day conditions and others bloom in short day conditions. To give you an example, you have an iris, which is on the left, and a chrysanthemum, which is on the right. That pink flower is a chrysanthemum. Okay? I'm going to use those two flowers as examples as we go through this. So while we call them long day and short day plants, we actually care less about how long the day is, because the critical factor is how long the night is. Granted, it makes sense to call them long day and short day because it is what it is, but we care about the night length more, which is obviously confusing. So that bottom line, the control of flowering is achieved through photoperiodism. That just means how long the light is available. Okay? Hopefully you're with me so far. Now we're really going to get into it. So the big thing with flowering plants is phytochromes. Phytochromes are pigments that are found in plants. They are used to measure the length of night. Remember, we call them long day and short day, but we really care about the night length. So we care about this because phytochromes change from phytochrome red to phytochrome far red. So from here on out, I'm going to refer to these as PR and PFR. So PR is phytochrome red, PFR, phytochrome far red. We care about both of them, but we care more about phytochrome far red, PFR. We care about this because that determines the critical level that plants are at to determine if they're going to bloom. So a quick overview of what happens during a day is PR is converted to PFR during the day. So you can see that here, that during the day you see PR with a bigger arrow being converted to PFR. It happens a lot and it happens quickly during the day. PFR slowly comes back to PR during the night. So you get a lot of PR from the sun and it's converted straight into PFR. As the night happens, PFR slowly goes back to PR. In daylight, there's a lot of red light, right? So the sun gives a lot of PR and not much PFR. In darkness, you have neither. So again, in the sun, plants get a lot of PR, convert it to PFR. At night, PFR slowly becomes PR again. So this is kind of a verbal, I gave a verbal explanation of what was going on, and this is it written down. So again, PR is absorbed through the day, converts itself into PFR. So if you have a long day with a lot of sunlight, you're going to end up with a lot of PFR at the end. If you have a short day with less sunlight, then not as much PR gets converted to PFR because you don't have as many sun hours. And at night, the opposite happens. So PFR converts back into PR. So long day plants will only flower when the night is short. This causes a lot of PFR because the day has a lot of PR being converted to PFR. So you end up with a lot of PFR. When PFR is allowed to build up to a critical level, it stimulates the flowering hormone. Once that hormone is released, the flower will bloom. In short day plants, you have the opposite. So you have really long nights, which means that you have less PFR. The day is not as long, so less PR is converted to PFR, and the PFR over this long night is going back to PR. So once the PFR level falls below a critical level, the flowering hormone is released and the plant will bloom. Recap. You have long day plants. Long day plants have a lot of sun, less night, right? Nights are short. Because the nights are short, less PFR is converted back into PR. If you have high levels of PFR, that means that the hormone will be released and the iris will bloom. Short day plants. Nights are long, which means more PFR is converted back into PR. Once the PFR levels are low enough and they drop below the critical level, then the flowering hormone is released and the chrysanthemum will bloom. They will not be able to bloom in each other's time frame, right? Like winter versus summer or spring versus fall, because the amount of PFR is critical to determine if they will bloom or not. That is the last bit of this unit. This is the last lecture, and this is the last slide. If this last bit, this flowering, is still a little questionable, I do want you to re-watch this and also check out some of the videos on the Google site so that you have a better idea of what's happening. I'll see you next time.